Okay, I shall begin. Could you confirm that you are seeing my screen and I'm audible? Can you hear me? Okay, sure. Fine. Okay, so I'll begin. All right. Okay, so welcome everyone. Thank you for joining for the problem solving session 3. And we will continue from where we left last week, a small portion which was left last week, which I think is important. One of the important practices in proteomics, you should know that. And then we will switch to enzymes. If you have seen the lecture notes for week 3 and 4, they are both enzymes like overlapping. So what I will do is first cover the week 3 enzymes today. So the partial Michaelis-Menten equation and a lot of numericals which show how to use that equation and solve numericals. And in the next week, we'll do the inhibition, competitive inhibition and all the inhibition with this, which regard to enzymatic kinetics. So, but today we will first begin with the techniques. We'll cover it up, we'll finish it, and then we'll move to enzymes. Okay, so yeah. If you remember last time we had finished at SDS page, we looked at what is SDS page, and if you have SDS page profile, how do you calculate the or how do you come to the size of the fragment or protein that you are looking at. The other technique is chromatography. Before I begin to explain what is chromatography, there is this question that is also there was there in the syllabus assignment last time. So before I start talking, could you look into the options and tell me which ones are correct? So the question is, basically you need to identify true and false statements and then give me the ratio of false to true statements. So out of the given questions, out of the given option, which ones are true, which ones are false, and then give me the ratio. Basically just identify the number of false sequences here. So these are a set of 10 questions, 10 statements, and relating to chromatography and SDS page. So it will give you an understanding of both the techniques. So let me know when you are like, I will discuss the answers after a few minutes. Let me read out the sentences for you. So the first one says, chromatographic techniques are preparatory in nature, whereas electrophoretic techniques are analytical. If you're not sure what the terms are, we'll explain it in a while. Next, although DNA molecules are usually separated by PAGE or polyacrylamide gel electrophoresis, agarose gel electrophoresis is used to separate DNA oligos generated during sequencing by synthesis methods. C. TEMET or tetramethylethylene is used as a catalyst for the polymerization of acrylamide during gel preparation of SDS page. Next, in agarose gel electrophoresis, gel formation is due to chemical reaction, while in PAGE, gel formation is due to hydration. Next, the running buffer in agarose gel electrophoresis is TAE or tris acetate EDTA buffer, whereas the running buffer in SDS page is tris glycine. Next, the gel in SDS page is thick to prevent the aerial oxidation of acrylamide to acrylic acid. Next, ampholites are used to establish an electric potential gradient for isoelectric focusing. Next, proteomic studies often make use of 2-day page. The last two are in laminalized discontinuous page. The stacking gel has larger pore size than the separating gel, and glycerol is added to proteins to ensure they sink to the bottom of the well created in page. So how many of these do you think are correct? How many of these do you think are incorrect? And then give me the ratio of it. You could simply write the number of false sequences. That should be okay. That should be the same answer. So what are the how many of these sentences are false? I see people are typing, so then we'll discuss after we receive the answers. A, B, D, F are false. A, D, F, J are false. Okay. D, E, J are false statements. Okay. All right. We'll start discussing the answers. So the expected answers of false sequences is 4. So 4 by 6 is 0.67. That's how you need to report this value. So the false statements are A, B, D, and F, as someone I think had written. So I think most of you did write A, D, F. So let me explain each of these statements one by one. So the first one, which is stating that chromatographic techniques are preparatory in nature, while electrophoretic techniques are analytical in nature. So if you understand the basic difference, that this should be easy. So preparatory chromatography or preparatory techniques in general is used to isolate and purify compounds in large amount. So you basically prepare the sample, so you isolate the sample or purify this compound. While when you're saying analytical, what you're doing is you're analyzing. So you're identifying the proteins, you're identifying the compounds, you're quantifying. So if you look at chromatography, generally, it might seem to you that it is used for isolating and purifying, but you should also know that there is analytical chromatography as well, which is like thin layer chromatography, TLC. So you can see what kind of composition is present and how much of that are present. So or gas chromatography, which you can use to identify which kind of molecule is present. Whereas electrophoretic, it seems like that they will be analytical, but there are also some preparatory electrophoresis techniques. So both these techniques can have electron for analytical and preparatory use. So that's why this is a false statement, because here it categorically says that one is used for preparatory while the other is simply used for analytical. That's not correct. That's why it's a false statement. Next, it says, although DNA molecules are separated by page, agarose gel electrophoresis is used to separate DNA oligos generated by sequencing by synthesis. So, agarose gel electrophoresis is the technique which you do after PCR. So, when you are doing PCR, the gel that you run to check your PCR product confirmation is agarose gel electrophoresis, right? Now, sequencing by synthesis is basically if you're trying to generate oligos. Oligos are basically nucleotides. So you're synthesizing nucleotides. They can be very small in size also. So for a smaller resolution, we generally use page and not agarose gel electrophoresis. Agarose gel electrophoresis is a good matrix to resolve from 100 base pairs to a kilo base pair of fragments. But if you want to do sequencing by synthesis, which are smaller fragments, we generally do page. So it's actually written the opposite. You are using DNA molecules for agarose gel electrophoresis, but you are using the DNA oligos, which are shorter in size, which are usually separated by page. So that's why this is a wrong answer. Now C, which is TEMED, is used as catalyst for polymerization of agarose acrylamide in SDS page, is a correct statement. Where if you prepare SDS page, so basically there are components. The matrix page, polyacrylamide gel electrophoresis, is basically a matrix formed due to polymerization of acrylamide and bisacrylamide. So that polymerization is taken or is felicitated by TEMED. So this is correct statement. Next is in agarose gel electrophoresis, gel formation is due to chemical reaction, while in page it is due to hydration. Now let me explain you the way these two gel are made. So in agarose gel, what we do is we take a certain amount of buffer and we take certain amount of agarose. Say if we are trying to make a gel of 1% agarose gel for our experiment, we'll weigh 1 gram of agarose powder and we'll dissolve it in 100 ml of buffer. So we are dissolving it by heating it. So we heat the container, we heat the flask. In the agarose dissolves in the buffer, and we pour it with our fluorescent dye, and we pour it into the matrix, and we let it solidify. That solidifying is happening because the solution is getting cooled down, and there is hydration. While in page, in polyacrylamide gel electrophoresis, what is happening is we have acrylamide, we have base acrylamide. We are not heating, we are putting TEMED. We have APS, we have TEMED, we put all of these so that we have polymerization of base acrylamide and acrylamide. So here, the gel is formed due to a chemical reaction, while in the first, which was agarose, you have cooling down of the solution, which is leading to formation of the gel. So as they cool, because of hydration, these molecules are coming together and they form a matrix. So it's also written in the opposite way. So agarose is what is formed by hydration, like agarose gel electrophoresis, while in PAGE it's formed due to a chemical reaction. Next, it says running buffer in agarose is tris acetate EDTA. It can also be tris borate EDTA, TBA or TBE or TAE, while the running buffer in SDS PAGE is tris glycine. So that's correct. Next is the gel in SDS PAGE is thick to prevent the aerial oxidation of acrylamide to acrylic acid. This is wrong because we don't, the thickness of the gel is not because of you're trying to prevent acrylamide from getting oxidized. First of all, it is stemmed, which leads to polymerization of acrylamide. And what is the polymerization of? The polymerization is of free radicals. So it's not acrylamide turning to acrylic acid, is the free radical that gets oxidized. And for preventing that, we are using APS, ammonium persulfate. But thickness has nothing to do with preventing oxidation. Thickness is basically how well do you want the conductivity to be through the gel. So thickness is a different parameter, and you trying to prevent oxidation is a different parameter. So that's why this is false statement. The rest of the four are correct statement, where it says that ampholites are used to establish an electrical gradient, which is correct because ampholites are basically those that have both negative and positive charges. So they create the potential gradient. Proteomics, you use 2D page. So 2D page is basically as similar in as to the point above, where you have this isoelectric focusing because of the potential gradient, and then you have electrophoresis based on size. And the second last one says that lamellized discontinuous page is basically two gel. The first is stacking gel. So basically, as the name suggests, you have a stacking part and then you have a resolving part. In the stacking part, what you are trying is you want all the proteins to form thin bands in their respective wells so that they all get to the bottom of the well so that they can disperse. So all of them move together. And then you have the resolving gel, which, as the name suggests, these proteins now resolve according to their molecular weight. So discontinuous because you have a stacking and then a resolving gel. And the last one says that glycerol is added to protein samples to ensure they sink to the bottom of the wells. That's because your protein needs to have certain weight, because when you are, so SDS page is generally vertical. So when you want your proteins to slide down through the gel, you want it first to reach to the bottom of the well so that it start moving through the pore of the gel. So for letting it sink down, you need to make it heavier, and you put glycerol so that it sinks and goes to the bottom of the well. So that's why this statement is correct. So yeah, that explains this answer, and I saw that most of you did get the right answer, barring some statement, but that's okay. That just gives you an what do you say understanding of chromatography and SDS page techniques together. So quickly we'll do chromatography. So chromatography are basically separation of different molecules. As you can see here, you have column chromatography. So you have these column which lets you separate the different components of protein. It's a way, it's a method of separation of the different elements, different components into several parts based on how they are traveling through the matrix. Now, chromatography are of three types based on what are you using to separate those components out. You can be using ion exchange chromatography. You could be using, as we will see later, you could be using the other two methods that I will discuss. So in the first one, which you see here, ion exchange chromatography, it's basically using the difference in the magnitude and the sign. Sign basically means either positive or negative electric charges. So you're using that characteristic of the proteins to separate them, or any compound to separate them. So what you do is this column matrix is a polymer, and this has several bound groups. If the bound groups are anionic in nature, they are called cation exchanger. Why is that? Because anions have negative group, so they will bind to cations. So they will put the cations to bound to the column, and the remaining anions or negatively charged components will all move out through the column. Whereas if it's opposite, if the polymer, or if the column has cationic group, which is positively charged group. So as you can understand, if this is positive, it will bound to negative charged compounds. All the elements which are negative will stay bound to the column, and all the similar charges compound will move out into the effluent. So you basically are exchanging the cations, and that is leading you to separate your negative and positively charged components. So if you are using charge basis separation, that is ion exchange chromatography. Next is size exclusion chromatography. And this is easier. This is what we are doing in electrophoresis as well. But there, so when I say what we are doing in electrophoresis, what I'm trying to say here is in electrophoresis also we are using the size as a distinguishing feature. Based on the size, the, let's say, DNA moves, and the larger ones stay on top, and the smaller ones move faster, and we can separate the larger and the smaller bands. Here, what we do is we have again a column matrix, which is again cross-linked polymer. So they have different pore size. They have pore sizes. We can select the pore sizes. What we'll do is, if we have larger proteins, they will migrate faster. Smaller ones get trapped here. If they are smaller, they move and get stuck. The larger one can move through the side and then move out. So the smaller protein, they enter the pore here, and they get slowed because they have to now so they enter a pore, they get stuck. So they are mostly stuck. The larger one, which are not able to enter the pore, they simply slide through and go out into the effluent. So size exclusion is basically removing the smaller proteins and letting the larger ones move out. And lastly, you have affinity chromatography, which is simpler to understand. Here, what you are trying is you are trying to exploit the affinity between a ligand and the protein of interest. So let's say you have a protein in a solution, and you know that it binds to a certain ligand. So in your polymer, in your column, you make use of those ligand. You know the ligand will be there, the protein will bind to it, the protein will stay in the polymer, in the matrix, and the effluents and whatever comes out as elute will not have our protein of interest. We can later, once all the effluent is out, we can remove the eluent, and then we can use the matrix again to using buffer. We can reverse this affinity, and then we can elute our protein out as well. So you use the affinity between the ligand and the protein of interest to purify the protein or to get the protein out. So here you have these different chromatography techniques and a comparison of how does these from a total extract, how much of protein can you recover from these different techniques. So let's say you are extracting cellular content. So from that you have let's say 1.4 or 1400 ml fraction. So you get a 1400 ml solution. And you know there is 10,000 mg. This is all hypothetical. So imagine that there are 10,000 mg protein that you have recovered, and their activity is 1 lakh, and specific activity is basically total activity by total number of proteins. So specific activity is 10. Now, as you change your method of purification, your amount of protein that you recover changes, and the total activity, because they are purified, so the activity is also very high. So your specific activity also changes. So as you can see, moving from ion exchange to size to affinity, your fraction of volume that you elute gets diminished or reduces, but your specific activity increases because the more better techniques you are using, even though you are eluting less volume, you have pure compounds, so their activity is very high, and that's why the specific activity is too high. So depending on what kind of application you are using it for, that determines what kind of method you're using for purification. If you want to elute more number of proteins, then you go for something like ion exchange, where you have all the negatives or all the positives out. But if you want something very, very specific, then you use affinity chromatography, because you want only the protein of interest to be out in the like purified. So you use affinity, which has a less number of ml, but your protein content will be less, but you have the specific protein that you need. That's why the activity will be too high. So that determines, out of all these different methods, which method would you prefer to use for purification. Okay, now we have discussed all these different types of chromatography and what are they using for. So let's try to attempt this question, and this should give us an idea of what we are trying to explain and understand here. So there are 6 polypeptides. Look at the amino acid, look at the length of the polypeptide, see the charge on the amino acids and what are they charged. Overall, what is the polypeptide charge? So when mixtures of these polypeptides are subjected to the following chromatography techniques, separate experiment, uh, what do you think? So which of these peptides will come out first? So how do you answer that? Let's say all these 6 polypeptides are together in a solution. First, I put this solution in ion exchange chromatography, which has polymer beads coated with positively charged functional group. So which polypeptide will elute first? So which one will come out first? For A experiment, which one will come out first? That is the first answer. Similarly for B, now the opposite. I am doing ion exchange, but now I am using polymer beads which are coated with negative charge. So you give me the answer when I do negatively charged ion exchange chromatography, which polypeptide elutes fast. Think properly what gets stuck in the matrix. C is size exclusion chromatography. Again, think which size retains and which actually elutes out. And the last one is affinity chromatography with polymers carrying moieties that bind with arginine. So if your polypeptide has arginine, the polymer binds to it. Now, if that is happening, which one elutes faster? So give me the order of the polypeptide which will come out in these respective experiments. Take your time and think. Yeah, 4. I can see one answer. So take your time. Give me which one comes out first and in which one. Okay, 0136. We'll just wait if I see anyone else also trying to answer. Just we'll wait for 1 minute for other people also to see and look into the one question and see if they are able to identify the polypeptides that will come first. Okay, 6, 5, 3. All right. So I have a different set of answers. 6153 4136. See someone typing, so I'll come back and check the answers. I think we have already done this question in a previous. Yeah, so if I have, then we'll just quickly go over it. It was just in continuation of this topic, so I think we'll just quickly do with this. These were already in my previous slides in continuation of the previous topic. So if we have done this, this is a good revision for you. If people have not done this, this is for them. But we'll quickly go over it and we'll move to enzymes, for which we have a lot of numericals. Yeah, I think I have received answers. So let me see. People have already given me answers. And in case this was a revision for you and you got it right, so that means we are doing things correct. And if people are still thinking and couldn't answer, let me just tell you what are we trying to do here. Okay, so let's look at it. Let's first just look at the polypeptide and see what's actually happening. So what I did in the first one, or every polypeptide here is just showing you what is of importance to us. So in the first, I have shown you the positive and negative charge wherever it is present. In the second, third, fourth, wherever plus, it is a positively charged amino acid. Whenever negative, is a negatively charged amino acid. Wherever it is red, I am showing you glycine. Wherever it is green, I am showing you arginine. So now let's look into the first one. The first one says you have ion exchange chromatography with polymer beads coated with positively charged. So which one comes out faster? You can think in terms of if positive charge polymer bead, then the negatively charged polypeptide will get stuck to the matrix. So which one will elute faster? If it's a positive bead, the polypeptide which has the highest positive charge will elute faster. So if you look into the given polypeptide, which one has the highest number of positive amino acid charges, it is fourth. So you can see number of plus here is higher. So that's why this will come out, elute the fastest. Now, if you think in the opposite term, if you have negative charge functional groups in the polymer, then the ones with the most negative charge will come out faster. That's one, if you see here, right? Now, in the C, which is size exclusion, we are basically trying to understand which one comes out faster. And we have said the larger the polypeptide, the sooner it comes out. We have larger peptide here as 3 and 6. And if you now see 3 and 5, if you now see 5 and 3, so glycine and tryptophan are at the extreme end. Glycine is the smallest and tryptophan is the largest. Tryptophan and all these groups have this aromatic group, so they make it a larger group. Arginine is a bulkier group. So the more bulkier groups in the amino acid, the larger the protein. So you have the third one, which comes out faster because fifth one has more of glycine. So that makes it still smaller. So the third one comes out. And the sixth one, which is saying that it has a polymer which has interaction affinities with arginine. So we have 6. We have discussed it already, as someone reminded correctly, that if you now look at clearly, 2 and 6 are basically the same polypeptide. So the sixth one, which has more of sorry, which one has arginine, it will come out. So it will stay back. All of these has arginine. 2 and 6 don't have. So you can write 2 or 6. It's basically the same answers. So yeah, if people have already looked into it, we are just revising it once again. And now we'll move to the next question, which is a very, very simple question. So you basically have to separate lysine from this mixture of glycine, alanine, leucine, tryptophan, and lysine. So if you want to separate lysine, which one of these moiety will you use? So what is lysine charge? You think first that, and which respective charge will you use? And the answer is like, it is already saying you that we are using ion exchange chromatography. So which one will you use? I mean, which all will you use? They all have equal proportion of positive and negative polymers options. So if you are supposed to, by ion exchange chromatography, you're supposed to elute lysine later or purify lysine, which of these chemical moieties will you use? Which of these ions will you coat your polymer with? It's an easy question, shouldn't take much time. Yes, first answer. Anyone else? Okay, let me ask you this way. What is the charge of lysine? What amino acid? So anyone, so positive, right? So if I am doing ion exchange chromatography, what charge should be on my polymer in the matrix, basically? Negative, right? So your polymer should be negative. Your protein is positive. Your amino acid is positive. Yes, BCE. It makes sense, right? One more question before I actually will answer. In this setup, if you are trying to use a negative charge polymer, what type of ion exchange chromatography is this? An ion exchanger or cation exchanger? The beads on the polymer is negatively charged. So it is a think it is a cation exchanger or is it an anion exchanger? Yes. Anion exchanger, why? It's a cation exchanger, right? That's why I wanted you to confirm. Okay. So the exchange, the name that you're giving, I will go back to the slide. So answer, let me reveal the answer. B, C, E, as some of you got right. And I saw confusion. So if you see B, C, E, they have negative charges. If you see A, D, F, they have positive charges. Now let me go back to that slide quickly so that I can tell you what's exactly happening here. Those with bound anionic groups, anions are negative, they are called cation exchanger. Those which are bound with cationic groups are called anion exchanger. So how do you remember? Just remember whatever the polymer is binding to. If the polymer is negative, it will bind to positive charge amino acids. It is a cation exchanger. It is binding to cation, so it is a cation exchanger. If it is binding to a negatively charged component, that means the polymer is positively charged, so it is an anion exchanger. Don't get confused in this. I knew this confusion could arise. That's why I asked the specific question. If there is any confusion, please let me know, or else I will go ahead. Yeah, is this answer clear? Because I did see some people answering something else, but is it clear? We have this next question, but if the previous question is not clear, do put it in the chat box. I'll keep coming to this. If you have any doubt, please put in the chat box. I will keep visiting the chat box again. Okay. This question here, this says, okay, in biotechnological application, when a recombinant protein is to be expressed in a host and then purified, the plasmid containing the gene of interest is often flanked by a series of 6 to 9 codons encoding for a certain amino acid either on the 5' or the 3' of the gene, forming a tag. So you can see your tag here. This is tag. This is what will help you take out your protein out from the vector. So to obtain the recombinant protein, the cell lysate is subjected to suitable treatment, and the protein of the cell are separated from the other biomolecule. This mixture of protein is then passed through a column containing agarose beads coated with coordination complex of nickel and nitriloacetic acid. Imidazole rings there have an intrinsic ability to interact with this complex. This interaction is used to purify the recombinant protein and separate it from the other protein. Now, I've told you this technique. Now, let me know which kind of affinity it is, and the tag will have more of which kind of amino acid, histidine or proline. Yes, Aditi. Yes, lysine will stay back because that's the idea, right? That's what the entire 3 type of chromatography said. Whichever is the intended protein of interest, we design the polymer or we design the column to retain our protein of interest and let all the other move out as element. Be it my ion exchange, be it my affinity, be it my size exclusion. B and C, B and C. Okay. Okay. C and B and C, anyone else? So yeah, there will be 2 answers to it. One, I'm trying to know the type of chromatography, and second, I'm trying to understand what is the nature of the tag. For the tag nature, see what is given, which one it is interacting with, that is also given, and what is exactly interacting is also given. So let me know. So someone has sent affinity, which is A and C, and B and C. Okay. Anyone has any other answer? Aditi, was that clear? If it was clear, go ahead. Okay. Chat box is open for any kind of doubt here. Okay. Someone wrote ion exchange, I believe. Yes, let me also tell you why is it. So it is affinity because it is using the property of binding. You have a specific protein of interest, and then you have a tag in this which is identifying your protein of interest, and you have used something which binds to this protein of interest. You have a ligand here. This ligand is a coordination complex which has NTA and nickel 2 plus, which is binding to your protein. So you're using affinity, even though it shows that it is a plus charge, but remember it's a coordination complex. So it's not just an ion, it's a compound which has nickel 2 plus bound to it. So it's a complex. So you have affinity chromatography. Now it said imidazole ring. If you know histidine has imidazole rings, and it very clearly says imidazole ring has an intrinsic ability to interact with this complex. That means it has to have histidine in its tag. So the answer is A and C. Okay, hope this is clear so that we can move to this week's topic. This week the topic was on enzymes and Michaelis-Menten's equation. So let me know. Because now we're going to switch to enzyme. So if chromatography and an SDS page is clear to you, then we can start with this new topic. Okay. Let me start. All right. So enzymes basically, as we all know, they are proteins and they catalyze the hundreds of stepwise reactions for any kind of chemical reaction, be it degradation of neutral molecules, conservation or transformation of chemical energy, making biomacromolecules from precursor. All these you will mostly find while you are studying in some pathways, in some digestion process, anything that you read of any biological system, you will see enzymes being used. Not only in biological system, any system which is involving reaction, chemical reaction, you will see enzymes being used there, right? Now, these are protein whose catalytic activity depends on the integrity of native protein conformation. Most of the times you will listen that, or you will hear that enzymes are optimal, enzymes are very specific, enzyme work in a certain condition. That's because the protein conformation, which is in their native form, is integral to the functioning of enzyme. Enzymes are catalyzing reaction. For that, their 3D structure has to be conserved. Their 3D structure is of utmost importance. So anytime you're extracting protein, anytime you're working with enzyme, it is of utmost importance that you take care of the working, because you need to ensure that that stays in their native conformation, right? Now, enzymes sometimes also require these additional chemical factors which are important for their activity. So they need that cofactor, they need those additional components for their activity. For example, if they require cofactors as chemical components, just ions, for example, magnesium ions, manganese, or iron, these ions are called cofactors. But instead of inorganic components, if they have organic or metallo-organic molecule, they are called coenzyme. So coenzyme are by organic molecule. If you have only ions, they are cofactors. So they might require this cofactor and coenzyme for their activity. So here you can see these tables are taken from Leninger. Like I always say, I'm using the material from Leninger. So you can see this table shows you which enzymes are using which kind of cofactors. Some of the enzymes that you can visibly see and identify, if you have seen the pathways, you will know cytochrome oxidase, hexokinase. Can you guess? Can you tell me where is hexokinase used? Where have you read about hexokinase enzyme? If you know the pathway which is using hexokinase or pyruvate kinase. Glycolysis, exactly. One of the pathways that we definitely started reading with, glycolysis. So you have urease, dinitrogenase, arginase, all these enzymes, they have their chemical reaction that they facilitate. But for their activity, they require these cofactors. Similarly, some of the enzymes require coenzyme A, like so NAD, FAD. You will read in different pathways that these are components that are required for the enzyme to function. Enzymes, based on their activity, are classified generally into 6 classes based on their activity. The name says what kind of activity they perform. If they are taking part in redox reactions, they are called oxidoreductase. If you are transferring one group of, like there is a group which is attached to one component that transfers a part of this group to functional group to another component, that's transferase. Hydrolase, lyase, isomerase basically is involved in changing the structure, and ligases are basically attaching two components together. So you will mostly see DNA ligases, you will mostly see redox chemical reaction wherever you see. You will have isomerases, transferases, all these enzymes, you will mostly find them in different pathways when you are reading them. So these are the different type of enzyme. Now, if you have cofactors and coenzyme, then how the enzymes are defined is, you have these coenzymes are basically these cofactors or the coenzyme that you know of, so that they are tightly bound to the or covalently bound to the enzyme group. So they are called the prosthetic group. Prosthetic means something additional. So prosthetic group. Without the coenzyme, the protein is called apoenzyme. When they come together, they are called the holoenzyme. Holo means complete. So a complete catalytically active enzyme, which is bound with its required coenzyme or metal ion, is called a holo enzyme. So you have apo enzyme, holo enzyme, and its cofactor. Now, so why do you need this enzyme? What is the importance? So in nature, or if you just observe around you, there are many reactions taking place, but they are very, very slow in nature. So enzymes are basically used or basically there to catalyze these reactions. They help in occurring of this reaction faster. So enzyme provides a specific environment within which the reaction occurs rapidly. The reaction otherwise takes a long time to occur. Enzyme fastens this process, as you can see here. So you have an enzyme. This is a substrate that needs to be converted to product. What is the product? It basically wants this bond here to be broken. This bond will break on its own, but for that thing to happen without the enzyme, it will take a long time. We will see why in the next slide. So it will take a long time to occur. But if you have an enzyme that can work on this, then this breaks into product much quicker. And it has, so how will it do? Enzymes will have these sites where the substrates can fit. If you know, enzyme has active site which properly fits the substrate into it, and that gives it the specificity. So an enzyme is very specific for the substrate, and this specificity is because of the orientation of its groups that are there, and that orientation comes from the 3D structure. That's why your 3D structure of the protein is very important. So your enzyme has a pocket active site where your substrate perfectly fits in, and that leads the substrate to undergo the reaction which results in forming the products. Okay, so this is a way how you represent the enzymatic reaction. E is enzyme, S is substrate. Them coming together forms the enzyme substrate complex. Then the substrate changes to a product, so EP complex, and then finally enzyme moves away and products also leave, which is finally giving you S to P conversion. Okay, now what exactly is happening and why is it making it quicker? I'm sure you have already seen it in the slide. Let me just quickly go over this because understanding this is important. If you have any doubt regarding any of the slide, put it in the chat box. I will try to answer them. Right. So like I said, catalysts enhance reaction, and they do this enhancing of reaction rates by something called activation energy lowering. So any reaction that has several steps involves the formation of transient chemical species. All these enzyme substrate complex, enzyme product complex are basically transient chemical species that will break away to give the final product. So because they are intermediates of the final processes, they are called reaction intermediates. Okay. Now, when you have several steps that occur in a reaction, the overall rate is determined by the step with the highest activation energy. Activation energy is basically to move from one state to another state, you need energy differences. The difference in the energy is called the activation energy. The one which has the highest activation energy is the lowest to take place, and that's, so if you have, let's say, three steps in a reaction, and reaction number 2 has the highest activation energy, it is the slowest step to take place. So that is a rate limiting step. No matter your first and third can be as high as quick to take place as possible. So let's try to understand. Let's say this is your substrate, this is a product. For the substrate to turn into product, it has to cross this energy barrier by first upgoing the hill, energy hill. So it has to first acquire that much energy that it can come back to the ground state, right, of product. So this difference is called the activation energy. It has to have this much energy change for converting to S to P. While the opposite, from product to sub, if it's a reversible reaction, it has even higher energy jump to make. So this energy jump is what is activation energy, and by in its own, such reactions in nature, if it is not catalyzed by enzymes, it takes a very long time. It's a very slow process. Free energy change on its own is long process. But if you have an enzyme, look where does the enzyme brings down the change to. If you have an enzyme substrate complex, enzyme makes the energy barrier very low. What was this big a climb has now just become this much in its nature, and it doesn't even have to worry about this change because you have an enzyme substrate complex, enzyme product complex, which quickly forms and dissipates into the product formation. So whatever was this size, this energy difference has now converted only to this. So that's how you can see it is transitioning between S to P has become very quick. Now, how is it doing this? How do the pocket fitting helps? So it's a very good example. It is in Leninger. You can see this example and try to imagine and understand it better. So this is the uncatalyzed reaction. I'm using terms which are pertinent to the topic here. So let's say this is a substrate and it is a stick which is to be broken into two. If you know first breaking stick, you have to bend it and then it will break away, right? So for this to happen without any enzyme, this is the activation energy difference. Now you have enzyme which will enhance this reaction, but how does the pocket of the enzyme look like? Does it bind to the substrate or does it bind to the transient product that is there? So if you bind the enzyme here to the substrate itself, in the native form of the substrate, it binds to this perfectly because, like enzyme and substrate have a lock and key fit. So if it fits itself perfectly, now imagine what is to be done. For you to break the product or break the substrate into product, you have to bend it. That's the only way you can break. It can't be stretched, it can't be pulled, it has to be bent. If you have an enzyme which perfectly fits the substrate, then the force you require now to bend it is even further, which is basically of no use. You can see the climb here. The original climb was here, the black portion, right? But now you have an ES, which is basically enzyme on substrate and which is fitted so properly that the activation energy now to break ES to P is even higher. Let me put the pointer on so that you can see what I'm trying to show here. So you have this enzyme substrate complex with an enzyme that perfectly fits the substrate itself. So for that thing to break into product, the activation energy is even higher than it was in the native substrate form. This enzyme will be of no use because enzymes are supposed to enhance the reaction rate, not reduce it further. So where do you put the enzyme? Which enzyme or how does the enzyme work? So if you, instead of putting an enzyme here, if you keep an enzyme which is fitting the reaction intermediate form, then this will help you, or this reduces the reaction rate because it now allows the transition state to fit better. It will fit in this, and because it can now allow the formation of transition state quickly, the product will form quicker. So that is what is trying to be shown here. The moment you put an enzyme which fits the transition state, the change in energy diminishes, and that is the whole idea here. So your reaction rate increases, nothing outside these changes. So neither your equilibrium changes, neither the number of substrate, number of product changes, neither the energy is given by enzyme. So let's say you have a energy barrier, and to reduce it, enzyme is giving some energy. This is also not happening because enzyme is not participating. Enzyme is not changing in its form. It is just giving the required space for the reaction to take place in a faster way by allowing the formation of transition state and quicker. So it's just catalyzing the reaction by enhancing the rate of the reaction. Okay. Right. Okay. So let's move to some theoretical question before we move on to the maths of enzymes. So you have 3 questions here. Right. So let's quickly answer this with, so for the first one says, which of the following statements are true for enzymes? They are fibrous proteins. Collagen is an enzyme. They're spherical and folded. Coenzymes are inorganic molecule. Remember the terms clearly. Enzyme with incomplete activity is called holoenzyme. Apoenzyme is just the polypeptide chain. Second question says coenzymes. So these are different kind of coenzymes and their activity. So you can see which one of these are true. And the third one says, which of the following statements are false? So first you try to answer and then I'll give the answer to it. Let me know which one are you answering for. So maybe we'll go like this. This is the first question, this is the second, and this is the third. So let me know which one are you answering for. I'm guessing the C and the F are for the first questions. Okay, so I think first question is there. Okay, let's do the second one, which is about coenzyme, and the third one, which are statements in general, which are false. A and D. Okay. A, C, D. Question 2. Okay. Three, none of the above. Are you so you're sure, right? You're trying to say that none of the above are false. Please read the question properly. It says which of these are false, and none of the above basically means that none of the answers, none of the statements are false. They are all correct. Yes. Yeah. So I think I'll keep repeating in all my sessions. Please be reading the questions carefully. This is for whichever exams you take this thing in. Read the questions properly. And I think I received most of the answers. So let me discuss this so that we can move ahead. Yeah. So let me see the answer for the first one that we have given. We have got answers like CF. Okay. Okay. CF, as most of you have answered already. So the first one says enzymes are fibrous proteins. No, enzymes are globular proteins. Second, collagen, which is there in your matrices, if you remember, ECMs. They are not enzymes, they are proteins. Enzymes are spherical and folded. Yes, folded for sure, because they have to be. Coenzymes are inorganic. Let me see if someone did answer that as this thing. Yes, enzymes are coenzymes. Please remember, coenzymes are organic. Inorganics are called cofactors because they are ions. So cofactors, remember that. Enzyme with incomplete activity is not holoenzyme. It is just apoenzyme. Sorry, with complete activity is called holoenzyme. Apoenzyme is just the polypeptide chain, which is basically the enzyme. Apoenzyme plus its cofactor is called the holoenzyme. Okay, next is it was just for you to see which ones are you getting right. The correct ones are A, C, D. Biotin helps in transfer of CO2. Niacin helps in transfer of hydride, and coenzyme in acyl group. Riboflavin is not for amino acids. That's the wrong answer for it. And then you have this ones which are all false. So let me tell you this. Okay, by the way, niacin is for NAD, which you for the transfer of NAD group, nicotinamide groups. So the third one, biological reaction occur mostly without enzyme. No, they need enzyme. They have to have enzyme if they want to do it in a sustainable way, not too slowly. Enzyme can decrease or increase the rate and change the equilibrium of the reaction. The statement is entirely wrong. First of all, it does not decrease the rate. If that decreases the rate, that defeats the entire purpose of that defeats the entire purpose of enzyme. It increases the rate and it does nothing else. It changes no equilibria. So that is totally wrong. Third is enzymes are not specific. Again, that is entirely defeating the purpose, because if it's non-specific, it will bind to anything and everything and then it will not work. If it's specific, only then it works properly and then it enhances the reaction rate. There can only be one substrate in any reaction. That's not correct. We will see in the next session, there are something called inhibition, competitive, non-competitive, that whole occurs because it's just one substrate that it can bind to. And can be multiple substrate that it can bind to. The enzyme can bind to multiple substrate. Next is there are only non-covalent interaction between enzymes and a substrate. Well, that's correct, but that can also have some covalent intermediates. That's why not entirely it is correct. I mean, there is not only non-covalent, you do have some covalent interactions as well. And the last one, enzyme provide the energy required to cross the inhibition. No, it doesn't provide anything. It just works by lowering the energy. It doesn't contribute to the energy by providing it. It is lowering the barrier. Okay, let's do this. These are different types of catalysis. I haven't focused on the different type, but if we discuss these questions, the type of catalysis should also be clear. So the first one is general acid-base catalysis. Second one is metal ion catalysis. And the third one is a type of enzyme catalyst, which if you read, then you will know which kind of catalysis is talking about. So let me know which ones are correct for each of them. And if you have any doubt, please put it in the chat box. I am on the screen where I can see the chat box. Question 1, D, E, F. D, E, F. Okay. Okay, lot of answers. D, E, F, B, D, F, C, F, A, C, D, D, F. Question 1, D and F. Okay. Question 2, A and C. A and C are false. Okay. Do take care that the second one says false statements, the first one says true statements, and the third one also says true statements. Question 3B. Okay. Okay. Someone is typing. Let's wait for that person. B and E. Question 3B. Question 3B. Okay. 3. Question B and C. All right. I think I've got most of the answers. If there are few, I don't know if there are people who are still answering, but you can see the answers. Okay. Okay, let me explain to you. The first one says general acid-base catalysis. So first one says the proton donor or acceptor is H2O. Well, let me see first people did answer it or no. Okay, so I think you did not answer, maybe most of you, and that's correct because H2O being the acceptor is a specific acid-base catalysis where you are using water as the proton acceptor or proton donor. But if you are talking about in general acid base, it doesn't have water, okay, as the proton donor. Next is involves the formation of stable intermediate. I think people have given it false, true. Let me ask you, if you have a stable intermediate, then how do you break the intermediate? Intermediates are not supposed to be stable. Transient intermediate, they can only be transient if they are unstable. That's why they will break away to give the product, which leads to, if you know, Gibbs energy being negative. That transient nature of it is because of that, because it will break away and give it free energy change will drive it to perform the product. If it is stable, that means it has already reached its stable form. It will not break away further. For it to do that, it requires the Gibbs energy change. If it is a stable compound, that means it again has to cross a barrier of energy. If it is stable, it is not stable. It is a transient intermediate. It's called transient because it is unstable. Think I saw many of you write B, which is not. In case you haven't written, then it's fairly fine. Next is the transient state. Intermediate is a stable charge structure. The second one is wrong. This is exactly why it is also wrong. D is forming the involves the formation of hydronium ion. This is false again, because here the hydronium ions are formed only in specific acid base where you have a donor or a proton which has H2O in the medium. But in general acid base, it does not have to have this water as the hydronium ion formation as a step. Next two, if E is correct, which is catalyst provides the proton, it's acid catalysis. The F says when the catalyst provides proton, it's called base catalysis. Basically, the same statement. So E is correct, which if it provides the proton, as we know, acid provides the proton, it's acid catalysis. Base accepts a proton, so it takes away the proton. So that is base catalysis, right? Okay, moving ahead. Next to metal ion catalysis, the catalysis or the enzymes that facilitate the enzyme, the reaction is via metal ions. So there, if it says metal ions in this reaction exist only in single oxidation state, which is not correct. It can exist in multiple oxidation state as well. For them to take place in the redox reactions, the whole idea of redox reaction is it can exist in multiple oxidation states. So this is false. Metal ions orient the substrate for a given reaction. That's correct, because if they that's helping them. These metal ions are basically stabilizing and positioning the substrate so that enzyme active sites can interact with the substrate in a much better way. So they are orienting the substrate. That's correct. Next is the metal ions stabilize the charge reaction transition state as the charge on them is lower compared to the amino acid side chain. Now, metal ions stabilize the transition state or the negative charge on the transition state. And if uh, they are stabilizing the negative charge and metal ions have positive charge, that means their charge is much higher than the charged reaction states intermediate. So their charge is always higher than these negative charges. So this one says this is lower, it's false. And the last one says participation in the oxidation reactions by irreversible changes. There is no irreversible change, because if there is irreversible, it's basically stable, it is done for, and that is not what exactly is the idea. It has to be reversible so that the product formation can take place. It can transition into the next step. So there is nothing stable here. So the false statements in the second questions are A, C, and D. The last one, it shows you that there is a substrate A and B, and then you have enzyme, and then you have the formation of this intermediate compound, which is enzyme and substrate, B in the medium, and then water is again used, and then water is giving the ultimate charges to take out the enzyme, and you have A and B. Okay. So here it says the new pathway for the reaction has a greater activation energy. Even without looking into this, you will know that this is not correct, because if it has a greater activation energy, the reaction will not take place at all if it is and compared to uncatalyzed reaction. The whole idea is reduction in activation energy. Second is the hydrolysis reaction does not take readily place. That's correct, because if it had to, it would have taken in the first place itself. It requires this intermediate form for that to take place. The hydrolysis to take place. And the third one says the amino acid side chain cofactor form non-covalent bond with substrate. That is correct, because it also forms covalent, but as we have seen, covalent forms is not the major weight forms. It has to be non-covalent, which gives it the transient nature. So I think most of you got the third one correct. I can see that in the chat box, but I hope others also made sense to you. Okay, so let's quickly move to the kinetics part. If you have any questions relating to it, put it in the chat box. Let's just quickly do these enzyme kinetics equations, and then we have a lot of numericals to solve, which gives you a good practice of Michaelis-Menten equation. This is for today's class. Next class, we'll do competitive inhibition, where we'll do more numericals. Okay, well, let's go ahead. So enzyme kinetics basically talks to you in the whole reaction of enzyme forming a product with the substrate in its initial condition. How does the concentration of substrate, how does the rate of reaction dynamically change with each other? So Michaelis and Menten tried to put this together in an equation form. So what equation they gave is this. So let's first see the first one. So E is enzyme, as I have already discussed. S is substrate. So enzyme to substrate forms with a rate of K1. But as you know, they are transient, so they can also go back. They can also dissociate and form E plus S again, which is basically K minus 1. If K1 is forward reaction, K minus 1 is the backward reaction. But after a certain amount of ES is formed and the activation energy is crossed, ES goes to E and P. So enzyme substrate breaks to form the enzyme as it is product, which is a changed form of substrate. That forward reaction rate is K2 and the dissociation rate is K minus 2. And Michaelis-Menten gave us a relation between the reaction rate and the concentration of the substrate. So V naught is basically the rate of the reaction, is basically V max into S, Km, which is a constant, plus S. So this reaction is very important. This reaction should always be clear, and you should always remember this equation. Okay. And on the left is the graph which depicts this. So you have your Km here, and you have, this is the velocity. What is the velocity? So velocity of the reaction, basically enzymes concentration and substrate concentration. More the enzyme, more the substrate, they should form complexes and they should give product. So it should ideally have been linear. The reaction should have increased, keep on increasing as you have more enzymes and more substrate. But that's not the case. This is the Vmax basically. We'll understand what does that say, but what you see is the nature of the reaction is always plateauing. It is always sigmoidal, means the reaction reaches a plateau after a certain point. It reaches a plateau which it can no longer increase the rate, and that's because all the enzyme substrate complex have formed, all the enzymes have been used. Okay, so you have this plateauing nature, and we'll see what does half Mmax and Km, we'll see in the next slide. So this equation here, let's try to derive this and let's try to formally understand what's exactly happening. This is what you need to go back and keep coming back and remembering this. So let's do this. E plus S giving ES, giving you E plus P. This E plus P giving dissociating going back to the formation of ES generally is not favorable. So you can ignore this while you are determining the equation. Now, first you need to know is what is the reaction velocity. If you remember in the beginning, we said the rate limiting step determines the rate of the this thing reaction. So rate limiting step is the one that takes the longest time to happen. And here, breaking of enzyme substrate, because enzyme and substrate are there in the media, they will definitely come into contact and form the complex. But for the complex to actually break and give the product and the enzyme separation is which is the limiting stage. So the rate limiting step for this reaction with one step reaction is K2. So when you do V0, when you look into the overall reaction rate, your V0 is basically the reaction rate K2 into the enzyme substrate, which is the substrate for it to work on, right? So the reactant here. So let's look into this. So the rate of this formation, ES formation, is basically K1 into the reactant. So K1 into reactant. What is reacting? What is reacting is the enzyme. ET is total enzyme that is there. So total enzyme minus the enzyme substrate complex gives you the free enzyme. So the free enzyme is what will combine with the substrate to give you the ES formation or ES, enzyme substrate complex. So you find out the rate of ES formation is basically the free enzymes available in S. Free enzyme is basically enzyme total minus the ES complex already formed. Rate of ES breakdown is the reverse. So you have all the ES that is formed that can break away. So K minus 1 into ES. You also have K2 ES. So your ES is also breaking down to give you E plus P. So this reaction is also breaking down ES. This reaction is also breaking down ES. So when you are looking into breakdown, you are basically taking both of these into account. So you have both the rates taken together. Now what do? There is something called steady state assumption where basically you say the formation and the breakdown are steady, like they are in steady state. The concentration does not change, they're in equilibrium. So at equilibrium, ES formation and ES breakdown are equal. The rate of forming ES and forming or breaking down the ES is basically the same. So when you do the equating, you put both this equation side by side. Then you solve it further by opening this bracket here. You open the bracket. Then what you do is you take this component this side and you take away all the K1s, Ks into one side and ES into other side. Then you keep ES on the left side and you form this. Basically, you just take the numerator here to a denominator form. And this is the final form that you get. So you have ES is equal to total enzyme into substrate divided by substrate plus this rate constants here. This rate constants, which is basically K2 plus K1. So this forward reaction divided by the dissociation constant is called the Michaelis constant. And this is Km. So when you substitute that Km into this part, it becomes Km plus S and numerator has Et plus Et into S. Okay, and this is ES here. Okay, so what do? You now say you have V0. So you have done K2 into Et. So ES is basically velocity of the reaction. So you have introduced K2 into ES. If you do K2 into ES is V2. So V0 is here and K2 is here. K2 into Et into S. Now K2 into ET basically means all the enzymes have been used up. All the enzymes will be used up when the reaction reaches its maximum rate. So that's why Vmax. So your Maclaurin-Benton equation is V naught is equal to Vmax into S divided by Km plus S. This is how you derive the equation. If you work it out once, it becomes easier for to understand. Now, what you can do additionally with this equation, you know that the overall reaction is basically Vmax by 2. If you want to look at half time reaction, which is basically Vmax by 2, you can simply take it this side, remove the Vmax, you see K half is equal to S plus Km by S. Simplifying this tells you that when V naught, or when the rate of the reaction is half the Vmax, which is basically 50% of the enzymes have been used, that time this Km is basically equal to the substrate concentration. This is very important. This you need to remember. And this translates something like this. So Km, you can find out the Km as being the substrate concentration when half of the enzyme have been used. This gives you this point here. So this is what ideal condition would have been. But when you actually work it out, it forms a plateau condition, right? Now, in this form, your calculation can get a bit messier. So there is something which makes it simpler to understand, which is called Linear-Weaver-Burke plot. What do you do? Straight line equation, if you remember, it says basically y is equal to mx plus c. So you bring this equation into a straight line equation by simply inverting the equation. So instead of v naught, you are writing 1 by v naught. The numerator becomes a denominator here and the vice versa. Now this is why you separate this plus into two components. So Km Vmax by S is one component, plus S being S by Vmax S is another component. Now you can very clearly see this 1 by S is a common factor here. So you have a 1 by S here. And this S basically gets cancelled here because of the presence of S here. So you can cancel it. It finally looks like this. 1 by V naught is equal to Km by Vx divided into x plus 1 by Vmax. If you convert this to y is equal to mx plus c, it becomes very clear. Y is 1 by V naught in the straight line. M is the slope, Km by Vmax. 1 by S is your x. So you have your x here. And this is your intercept. So this is your intercept 1 by Vmax. So when your x is equal to 0, you have this. When your y is equal to 0, this is the intercept at the other line. So this is 1 minus 1 by Km. This is thus the representation of Michaelis-Menton's equation in a straight line formation. And then you have two other phenomena to take care of. So when you have Km, we have already explained, this is Km. So when you have K2 as the rate limiting step, K2, because we have said the one which is called rate limiting is the slowest in terms of the others. So your K2 being very, very less than K1, your Km basically becomes K minus 1 by K1, which is basically the dissociation constant. So Kd is equal to Km here. Okay, so Kd is basically K minus 1 by 1. This you need to remember when you are talking of one step reaction, and K2 is the rate limiting step. And when this we have already explained that whatever is the rate limiting step gives you the maximum reaction rate. If it is a three-step reaction, let's say ES forms EP and then EP also forms ENP, it's not just ES directly breaking into ENP. And if K3 is the rate limiting step, then whichever is the rate limiting step, that gives you the rate of reaction. This is what we wanted to highlight here, right? And then there is another phenomena which is called Kcat, or catalytic efficiency, which is basically rate constant for that limiting step. The limiting step here, how do you find the rate limiting constant? When you do the calculations and you've come to the terms of Vmax by ET. We know Vmax is basically all the enzymes taken into the formation of S. So Kcat into ET. So K catalytic efficiency is basically Vmax by ET. This is called turnover number, which is equivalent to the number of substrate molecules converted to product given in a unit of time for a single enzyme when the enzyme is saturated. So when you're saturated, when all the enzymes are bound to substrate, at that point it's called saturated. And when you have that, how many substrate it can convert is what is referred to as a turnover. How many substrate has turnover to a product, so turnover number, right? And each enzyme has its own catalytic efficiency and Michaelis-Menten constant, which gives totally, it's the overall, it gives it's the specificity constant, right? Okay, so well, these are all equation derivation and a lot of terms that you need to know. So kcat, km, vmax, et, k2, all these you should remember, and with more numericals this will become clearer to you. Okay, let's do the first question here, which is let's try to understand what you've understood from the graph first, and then we'll do numericals. So the first one question here is in this set of statements, which one is true for Michaelis-Menten kinetics? So is the rate of the reaction directly proportional to the substrate concentration? Next, the reaction rate reaches Vmax after a long time. C, formation of the enzyme substrate complex takes a long time, but the conversion of complex to products is occurring quickly. D is the opposite. That means ES forms quickly, but the conversion of product forms takes time. E, saturation effect is, so when does the saturation takes place? Saturation effect is when there is excess enzyme to combine with the substrate, or the saturation is there is no free enzyme to combine with the substrate. And when Km is equal to X is the V naught, or basically the rate, Vmax by 3. Which one of these is true? D and F. Anyone else to answer? Okay, see, F is okay. F is correct. Okay, someone else is typing as well. A C E, A C E. Okay. Okay, let's do this quickly first. Okay, so let's look into this. Rate of reaction is directly proportional to substrate concentration. Let me go back to the equation here. It is not directly proportional to the S. The S component is also there in the numerator and the denominator. So it is not directly proportional to substrate concentration. It is, the form is Vmax S plus Km by S. So it's not directly proportional. That's why this is not correct. Reaction rate Vmax reaches at a long time, then the whole purpose of having an enzyme here doesn't work out. It reaches in a quicker time. So the formation of enzyme substrate complex takes a long time? No. Enzyme and substrate forms quickly because that's the whole idea. But what was the rate limiting step? K2, where your enzyme substrate breaks into enzyme plus product. This step here. Let me go back here. This was the rate limiting step. Again, let me show you here. This here, the reaction rate is given by K2ES. This is the rate limiting step here because this has the lowest time, or the longest time to happen. And saturation effect is when you have excess, if you have excess enzyme to combine with the substrate, then there is no saturation yet. Saturation means you have filled all these active sites on the enzymes. So you have no more enzyme sites available to substrate for binding to the enzyme. So the enzyme substrate happens when there is no free enzyme. When there is excess enzyme, it has still space to find. And last one, Km is equal to S. The Vmax, V naught there is half of Vmax. This is what the last statement here was. So it is half of Vmax. Okay. So these are the correct statements. And if you have any doubt, put it in this question. Okay, so we have 30 minutes for our numericals, which is a great thing because we get to practice a lot. Okay, let's do the first one here. Right. So the slope here is 0.5 and the area of triangle OAB is 0.125. I would like you to note down the values first because I would like to draw, keep you with the equation graph. You derive the answer on your own without looking at the options here, but do write it down. The area is 0.125 and slope is 0.5. I will let you be at this slide. make use of the information on this slide to give me the answer. The answer should be either for Km or Vmax. The first numerical here, let me again put it back to you. So you have a slope here. The line of the slope is given as 0.5. This is the Michaelis-Menten equation in straight line. It's called Lineweaver-Burke plot. The slope here is 0.5. And the area of this triangle here, we know this is 1 by Vmax. We know this is 1 as 1 by Km, is 0.125. And we have a set of answer, which is basically the values of Vmax and Km. So let's not look into the answer. We know we'll come to the answer as soon as we derive the or solve the equation. So if you know that, let's solve it here. So you have this value as 0.5. You have this, and this triangle's area as 0.125. So give me the value of Km and Vmax. Once again, you have your slope here, which is 0.5, and this area is 0.125. And you're supposed to solve it for Km and Vmax. If you solve one, you will get the other just by the same equation. So try to solve it for one, try to solve it for Km. You should land up with Vmax, or try to solve it with Vmax, you will end up with Km. You come to a solution, and then the solutions there are basically for the values. And if you are stuck anywhere, please let me know. I will definitely help you out. Vmax is 2 root 2. Can you give me the value? Should be easy. Root 2 is easy to find. Yes, if you give the values, then you will be able to look at the answer and give me the option value correctly. Because the answer options are in exact values. Let me show you the answer values first. Yeah, so they are in 1.41, 2.82. So do you have the values? 1.41. What is 1.41? I'm sorry, I'm taking those exact values because that matters for the answer. So is this Km or is this Vmax? Km is 1.41. Okay. Okay. So if Km is 1.41, what is your Vmax? 2.82. Basically. Okay. So now that you have both the answers with you, Km and Vmax, can you give me the answer for this question? So what is the answer to this question then? So Aditi, you got it right. 2 root 2 is basically 2.82. Square root 2 is 1.41. So the values were not there, but you reached the correct place. B. Okay. A. Okay. Who's left? A. Anjali, do you want to stick to your answer or do you want to change it? It says minus 1.41. It's okay if you are stuck with it, that's fine. I will anyway be asking you again. Okay. Anjali, while Anjali thinks, I'll give the answer and then wait for Anjali to tell me if she is agreeing or if she wants to change the answer. Sorry, my light is being absolutely dysfunctional today. Okay, while Anjali is typing response, few people have reached the correct place. So 1.41. And if your Km is 1.41 and Vmax is 2.82. So this option D automatically goes, or C probably goes wrong. D says minus 2.82, which is also not correct. E, because you are able to work with the values, so it's not non-real. Now the question is Km is 1.41 or Km is minus. As per the graph, Km is on negative side. That's why I put the well, the question here is about Km. So the answer will be in terms of Km, like whatever you solve for. You don't, so when you are solving for whatever equation you are getting, the answer you are getting is what you're supposed to put out here, right? Because even though it shows in the graph by equation, I'm sure you would have gotten 1.41. So you put that answer out here and not the negative one. Okay. And let me first give you the answer. Is this what you people did? Is this exactly what you did? Because I don't want to explain if you have already done this one. This is what you have followed for triangle area. This information we already had. So you just substituted one for the other and you got the answer. Is this what you did? Is this what you did already? I'm just confirming. So this is what you did, right? Yeah. Because I also have to upload this on the internet, so I have to explain this to the people who visit it on YouTube. So give me 30 seconds to explain it to them who if my class couldn't answer or will see me later. So you have a slope which is Km by Vmax, which is 0.5. I've written 0.5 as half so that it gives me the equation between or relation between Km and Vmax. So twice of Km is equal to Vmax. The other set of information I had was triangle OAB having 0.125. Because it's an area, I will use the positive values for the area here for the values for all the nodes. So half into base into height is the standard formula of triangle area. So I've substituted half. Base, this is base, so this is 1 by km instead of minus 1 by km, and height being 1 by Vmax, 0.125. I've basically written 0.125 by 1 by 8 because I don't want to deal in points. And that gives me this equation, which is 2 Vmax by 8 is equal to 1 by km. Then I have two equations which uses two variables, Vmax and km. I've substituted one for the other. So what I did is I substituted Vmax with km, and that brings me to this equation, 4 km by 8 is equal to 1 by km. When I further solved it, I solved it for km and I got to the value of 1.41. I already had the equation with me that Vmax is equal to twice of km. That led me to Vmax being 2.82. So that's how the answer here, as I had mentioned previously, what answer you get by solving is what you should point out here. Okay. All right. So let's move to the next set of questions. Now, an enzyme follows Michaelis-Menten's kinetics and with a substrate S. The fraction of the maximum velocity, which is what is the ratio of V naught, that will be observed when you have substrate, which is 4 km. So solve it for your V naught and then let me know what is the fraction which is being used, or what is the fraction that you will observe when your S is 4 km. You know the equation. You just need to substitute one value for the other. If you are confused with the equation, let me put you to this form, or maybe put you to the original form, this one. This, as I said, you need to remember. If you have noted it down, it's well and good. If you remember, that's perfect. If you are still finding it difficult, you can note it down for your reference and use this equation for your solving the answer. Your S is 4 km. So what is the fraction of Vmax that is being used currently in this question at substrate concentration being 4 times km? Four by 5 is equal to, okay. Yeah, I wanted to highlight this. Questions like this come in GATE, question like this come in your assignment and an exam, and they give you the cue of how to answer them. The cue here is correct to one decimal place. So your answer should be in the correct format. In case anyone of you gives GATE or CSIR or wants to, or you're taking any exam, please answer in the correct format. Correct. Now you're correct. So you're in decimal places. Right. Others? So your cue to numerical questions in what form you should answer are always there in the question. Read the question carefully. Once you are done solving, first look into the cue. What is it saying? Is it saying fraction? Is it saying integer? It is saying, if it is integer, you round it off and give it in an integer. If it is fraction, you write in the fraction form. If it is decimal, it means there will be decimal and you need to round it off to whatever it says. It says one decimal here, so you have the one decimal answer. Vmax is 1.25. Okay. 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 Anjali, exercise for you. The question asks which of this? Which one of your this is final answer for the question? Because here it is numerical question right now. So I want one number. So which number will you give me as the final answer? 0.8 or 1.25? And while she's doing that, others please feel free to give your answers here. Okay. So let me read the question once again. The question says the fraction of maximum velocity Vmax will be observed with the substrate concentration being S is equal to 4 km. So fraction is what I need. 0.8, another answer I have. Okay. Let me, as you people have already answered it correctly, 0.8. And this is what you have done. I am sure this is the question that you used. V naught is equal to Vmax into S divided by S plus Km. Where basically in place of S, we have to write 4 km. So 4 km in the numerator, 4 km in the denominator. In the numerator, it becomes Vmax into 4 km. In the denominator, it is 4 km plus 1 km, 5 km. Km gets cancelled. You have Vmax into 4 by 5. Now, yes, Anjali had answered already. So correct. So you are supposed to give me the fraction of Vmax that is being. So 4 by 5 into Vmax. So it means 4 by 5 of the fraction of Vmax. So the answer will be 0.8 as correctly written by all of you. Okay, next question. So an enzyme obeying Michaelis-Menten kinetics shows a reaction velocity of 10 micromole per minute when the substrate concentration is equal to Km. So the maximal velocity will be, for this enzyme will be, where will we find these recorded lectures? These will be, so you do get a link to the Excel sheet that gives you the YouTube lecture links as well as the PPTs. I am sure you would be shared with, that would be shared with you, right? You will have the recorded lectures in those link. So the lectures will be uploaded in YouTube and you can find those links there. Yeah, coming back to the question. So basically you have the you have Km being equal to S and you have the velocity. So what is the final velocity, the maximal velocity? 20 micromole per minute. Okay. Others? $20? Yeah, no problem. Okay. Shall I wait? Okay, we'll have other questions we'll do together. Right, so as people mentioned here, 20, which is basically substituting here, which is V naught is equal to Vmax S plus Km, where basically this Km becomes S. Vmax is basically, or Vmax S divided by 2S is basically 10, which is the value given to us. So Vmax becomes this 2 goes up there 2 into 10, if you remember this. Or you can simply remember the Vmax by 2, which we solved for looking at half the velocity when it is what is the condition there, and we know that Km is equal to S is basically Vmax by 2. So if Vmax by 2 is 10, Vmax is 20. You can use that logic or you can solve these equation and get to 20. Okay, yeah, next question. So for a given enzyme catalyzed reaction, the Michaelis constant is 0.6 millimole and the substrate concentration is 1 millimole. Again, what is the fractional saturation of enzyme under this condition? Same kind of question, same kind of logic. Okay, 0.625. Okay, others? I will wait because I think people are still solving it. We do have more numericals. If we are left with numericals in this class, because I don't want to break the continuity, and because next lecture will also be on enzymes, if you have looked into week 4 already, they are also enzymes. So I will have some of these questions so that it gives you a refresher of what we have done today, Michaelis Menten equation. And then we move to the inhibitions and different types of inhibitions. So if we are left with some question, which I think we will be, because we are already 8 minutes remaining. So we will do those questions in the next class, get a refresher of the exercise today, and then we'll go to the next new topics there. So it's okay. We could do one more question today and then we can wrap it up. But in the meanwhile, I see other are still trying to answer. So yeah, I'll wait. Right. Okay. Let's do this question. 0.625. And this is what we did, right? Now, before I go to this, you will also see 62.5%. You can also answer in this. You can also answer as this. Why is that? Because in my question here, I haven't specified the type of answer I need. So you can give me either in fractions or you can give me in percentage. But in the previous questions, let me just take quickly back. Every time I have given you questions in these two, I have told you what kind of answer I need. So be mindful of the questions requirement, right? So you 0.625 is also correct. If someone had written 62.5, that also I would have accepted. Or if someone would have given me any other percent or fraction that leads to 0.625, would have accepted that as well, which is basically 1 by 0.7. Yeah, 1 by 1.6. So you can give me that also, I would have accepted that. Okay, let's look into this. So we know this value and we know V naught is equal to K cat into ES, V max is equal to K cat into ET. Catalytic efficiency of total enzyme is basically giving you the final maximum velocity. At instantaneous ES is gives you the instantaneous velocity. What question wants me to do is, question wants me to find out the fractional saturation of enzyme. So out of the total enzyme, how many of these enzymes are saturated? Saturated is basically is bound to substrate complex. So I needed ES and ET. So I translate, or I use the equation which has ES and ET. So ES basically is related by this formula. ET is related by this formula. I need to find out the fraction of ES to ET. Basically, I need to find out the V naught by V max. If I take K cat, they will ultimately get cancelled. So V naught by Vmax, which is basically this reaction. I can just simply take Vmax from my right side to my left side. It becomes same as this. S is equal to Km plus S, which is 1 by 0.6 plus 1, which is 1 by 1.6, which is 0.625. Any confusion? And was this what you thought while you followed the answer? Yes, cool. Okay, I have only 4 minutes remaining, so we'll do one more question today and then remaining we'll do in the next class. So given an enzyme with Km of 0.5 millimole, at what substrate concentration will the velocity of the enzyme reach 1 by 4 of the Vmax? And Vmax is given to me as 200 nanomole per second. Basically, you need to give me the substrate concentration at instant where the Vmax will be 1, V naught is Vmax by 4, and I know the Vmax already. Vmax is 200 by 8. Wait a second. Where did you get 8 from? And are you asking me a question? Are you giving me the answer? I mean, is there a doubt there? So 1 by 4 of Vmax, why is it 8? It clearly says whatever my V naught is, it is Vmax by 4. And I am also giving you Vmax, which is 200. So it's 200 by 4. 0.167. Okay, your answer. Just a second, Jayakant. I will come back to you. Anjali, is this clear? Vmax is 200. V naught is Vmax by 4. So 200 by 4. Okay. So yeah, I've got one answer. Maybe use this information and answer quickly. What is the substrate concentration at these conditions with your V naught being 1/4 of Vmax and your Km being 0.5? .16. Okay. 0.16 or 0.17? Where will you place it, Aditi? Because I know you have the decimal places. So will it be 0.16 or 0.17? Well, it is. Okay, I'll tell you what I am trying to refer to. How many of you here have want to give gate or are thinking of giving gate or have already given gate? All of you have given gate already. Or any kind of national level exams, CSIR or gate or anything. You can give the hands up, right, given. So anyone and the others? Yeah, 1 by 6. So if you had planned to give gate or if you are giving CSIR, you would know or you would have seen or you will see in the question papers that you'll get a lot of questions on enzyme kinetics. And enzyme kinetics, the first few questions were of the pattern of gate paper, and they generally give you round it off to 1 decimal places. So when you do 0.167, if I ask you to give me only 2 decimal places, what will be the correct answer, Aditi? Will it be 0.16 or 0.17? Yeah. So I hope Jaykant also is clear on this, because if it's 0.167 and the answer question demands you to write in 2 decimal places, you have to round it off, it becomes 0.17. So 0.16 will be incorrect because your rounding off also takes it to 0.17, unless you want to write it as 0.167. When it is not cleared, when it is not specified, it's better to write it in at least 3 decimal places. And I can see most of you have got the correct answer. So yeah. So basically we have given all the information here. We have been given Vmax, we have been given Kf, and we also have, because we have Vmax, we can find out the instantaneous velocity, which is basically by 450. And now you just need to substitute. You have 50 here, you have 200 here, you have S here, which is what you need to find out, and you divide it by 0.5. So I basically am solving it for the entire equation. I get it S is equal to 1 by 6, which is 0.17, and which is what all of you have got as well. And the slide here shows you, if in case someone is still figuring it out, what I've done is basically I've just taken this and multiplied the denominator to the left hand side. So it becomes 50s. 50 into 0.5 is basically 25, and 200s remains on my numerator on the right. Then I solve it for S. So 25 is remaining on my left side. I take 50, that side it becomes negative. It becomes 25 is equal to 150 S. S becomes 25 by 150, 1 by 6 or 0.17. The explanation for 0.17 I've just given. If the answer in decimal is 0.166666, so when you are rounding it off, it becomes 0.17. Well, so like I guessed, we are already over time. So I don't want to keep you longer here. So we can meet next class. And we'll do few more numericals. Here, if you have observed, all the units were in the same range. We'll have some question which will ask you to convert one unit into another. So those kind of questions are also important because that makes you pay attention to the question. So they will do those exercises and then move on to the inhibition of enzymes. And then I think we have polysaccharides. So we will do that part also. We have already done polysaccharides in the first class as well. So we'll do a revision and we'll solve a lot of questions on that. Right? So if you have any doubt, please ask me. If not, then we meet each other in the next session. Thank you so much. Thank you. I hope that was helpful. Thank you. Thank you so much. Thank you. We will have more numericals in the next session. So please do join. We'll give you a good exercise. Thank you for joining. Thank you so much.